Good morning, everyone, and welcome. I'm delighted to have you here today. My name is Dr. S.K. Singh, and I hold a PhD in this field. Today, we'll be exploring the fascinating world of ultra-performance liquid chromatography, or UPLC for short. This presentation will cover the fundamentals of UPLC, its advantages, and how it's transforming analytical chemistry. I look forward to sharing insights and answering your questions along the way. Let's get started. Welcome to our introduction to UPLC. UPLC stands for Ultra Performance Liquid Chromatography, and it's an advanced chromatographic technique that actually evolved from traditional HPLC, or high-performance liquid chromatography. The key difference is that UPLC uses columns packed with very small particles, specifically less than 2 micrometers in size. This small particle size is critical because it allows for more efficient separation of compounds. Another important aspect is that UPLC operates at very high pressures, up to around 15,000 psi, so the system can push the solvent through these tiny particles quickly and effectively. Now, what does this mean for us? Well, UPLC provides higher resolution, meaning we can distinguish between compounds that are very close in structure more clearly. It also allows for much faster analysis times, which is a huge advantage in busy labs. Plus, it offers better sensitivity, so we can detect even trace amounts of substances with greater confidence. Overall, UPLC is a powerful tool that enhances our ability to analyze complex mixtures efficiently and accurately, fitting perfectly into our goal of improving analytical performance in the lab. Let's take a closer look at the principle of UPLC. It's primarily based on optimizing the Van Deemter equation, which helps us understand how to achieve better separation efficiency. One key aspect is the use of smaller particles in the stationary phase. Smaller particles create a reduced diffusion path, meaning molecules have less distance to travel, which improves resolution and speeds up analysis. Another important factor is applying high pressure. By increasing the pressure, we can push the mobile phase through the column more forcefully, which further enhances the efficiency of the separation process. Now, when it comes to how separation actually happens in UPLC, it's based on differences in properties like polarity, molecular size, and how molecules interact with the stationary phase. These interactions determine how quickly or slowly each compound moves through the column, allowing us to distinguish between different substances effectively. All of these elements, particle size, pressure, and interaction types, work together to make UPLC a powerful tool for rapid, high-resolution separations. Let's take a closer look at the instrumentation involved in UPLC. First, we have the solvent delivery system, which is essentially a high-pressure pump that moves the solvents through the system efficiently and reliably. Next, there's the sample injector, designed for low-volume injections to ensure precision and minimize sample waste. The core of the system is the UPLC column, which uses very small particles, around 1.7 micrometers, allowing for better separation and faster analysis. On the detection side, we can use various detectors like PDA, UV, or MS, depending on the specific application and sensitivity needed. The data acquisition system then collects and processes all this information, giving us detailed insights into our samples. Now, moving on to the advantages of UPLC. One of the biggest benefits is speed. UPLC can run analyses up to 10 times faster than traditional HPLC, which saves a lot of time in busy labs. It also offers higher sensitivity and resolution, meaning we can detect smaller differences and more closely related compounds. Plus, it uses less solvent, making it more environmentally friendly and cost-effective over the long run. The better peak capacity means we can separate more compounds in a single run, improving our overall efficiency. All these factors combined make UPLC not just faster, but also more cost-effective and reliable for high-quality analytical work. Now, let's take a closer look at the applications adopted in 2023, which is the latest revision we're working with. This year, we've introduced some key concepts that are shaping our approach to quality and validation. First, there's the life cycle approach, which emphasizes viewing the product development and manufacturing process as an ongoing journey rather than a one-time event. This helps us maintain control and ensure quality throughout the product's entire life cycle. Next, we have analytical quality by design, or this is about designing our analytical methods with a clear understanding of how they perform and how robust they are. It allows us to proactively identify and mitigate risks. Speaking of risks, risk-based validation is also a core component, meaning we prioritize validation efforts based on potential risks to product quality. On the emphasis side, method robustness is a critical focus. It's about ensuring our methods consistently produce reliable results. Continuous verification plays a role here, too, helping us monitor and confirm method performance over time. When it comes to pharmaceutical assays and impurities, the goal is to accurately measure the active components and detect any impurities that could impact safety or efficacy. Stability indicating methods are designed to assess how the product holds up over time under various conditions, giving us confidence in shelf life and storage requirements. Bioanalysis, particularly in plasma and serum, is essential for pharmacokinetic and bioequivalence studies, providing insights into how drugs behave in the body. Forced degradation studies are also important. They help us understand how products degrade under stress conditions, which informs stability and storage guidelines. And finally, all of these approaches feed into our quality control testing, ensuring that every batch meets our high standards before reaching patients. Overall, these applications reflect our commitment to a science-driven, risk-based approach that enhances product quality and patient safety. Let's now look at system suitability requirements, which are essential to ensure our analytical method is performing reliably. First, we consider the theoretical plates, which give us a measure of efficiency. Higher numbers typically indicate better separation. Then, there's the tailing factor, which helps us assess peak symmetry. Ideally, we want it close to 1 for accurate quantification. Resolution between peaks is crucial, especially when analyzing complex mixtures, as it tells us how well 2 peaks are separated. Next, we look at the % RSD of replicate injections. This statistic helps us understand the precision or reproducibility of our method. Lower RSD values mean more consistent results. Retention time consistency is another key point. Stable retention times ensure that our method is reliable over multiple runs. Moving on, the broader context is method validation parameters, which are outlined by ICHQ2 and other regulatory bodies. These include accuracy, which confirms we're measuring what we intend to; precision, covering both repeatability and intermediate precision to verify consistency across different conditions. Specificity is about ensuring our method can distinguish our analyte from other components. Linearity, with an R-squared value typically greater or equal to 0.99, indicates our method provides proportional responses over a range of concentrations. We also evaluate the limit of detection, or LOD, and limit of quantitation, or LOQ, to understand the smallest amounts we can reliably detect and quantify. The range defines the concentration interval over which the method is valid. Robustness tests how resilient our method is to small variations in conditions, ensuring stability in real-world applications. And finally, system suitability checks are performed regularly to confirm that all parameters are within acceptable limits before analysis, maintaining confidence in our results. Now, let's look at the regulatory expectations for UPLC. It's essential that we demonstrate reliability, ensuring our system produces consistent results over time. Reproducibility is equally important, meaning that under the same conditions, we should get the same outcomes every time. Data integrity is a cornerstone, making sure all data is accurate, complete, and trustworthy throughout the process. Additionally, validation is mandatory before any QC release, confirming that our methods are suitable for their intended purpose. It's also important to remember that all these practices need to align with existing GMP and GLP guidelines. These regulations provide the framework we must follow to ensure quality, safety, and compliance across our operations. Keeping these expectations in mind helps us maintain high standards and supports the overall integrity of our work. Let's talk about stability indicating methods, which are essential for regulatory submissions. These methods help us ensure that a drug's stability is accurately assessed over time. A key part of this process involves forced degradation studies. These studies expose the drug to various stress conditions such as acidic, basic, oxidative, thermal, and photolytic environments. The goal here is to intentionally degrade the drug to see how it breaks down. It's crucial that our methods can separate all degradation products from the parent compound. This separation is vital because it allows us to identify and quantify each component accurately. Overall, this ensures that our stability data is reliable and meets regulatory standards, which ultimately supports the safety and efficacy of the drug throughout its shelf life. Let's take a closer look at the comparison between UPLC and HPLC within the context of the method lifecycle approach outlined by ICH Q14. As you can see, this approach guides us through each stage: development, validation, transfer, and verification. One of the key advantages of UPLC is its ability to work within a defined design space, which means we can optimize and control the process more flexibly. This design space concept allows for ongoing adjustments without the need for reapproval, supporting a more efficient development process. Continuous monitoring is another critical aspect, especially with UPLC, as it enables real-time insights into product quality and process performance. This aligns perfectly with the modern need for agile and responsive manufacturing. Overall, adopting UPLC within this lifecycle approach offers greater flexibility in regulatory submissions. It helps streamline communication with regulatory bodies and demonstrates a robust, science-based understanding of the process from development through to verification. So, in essence, UPLC not only enhances analytical performance, but also aligns with a proactive, risk-based approach to pharmaceutical development and manufacturing, making it a powerful tool in today's regulated environment. So, to wrap things up, UPLC is a regulatory-accepted analytical technique, meaning it's recognized and approved by major health authorities. It complies with important guidelines like ICH Q2, R2, and Q14, as well as FDA and EMA standards, which ensures its reliability and acceptance in the industry. What's really great about UPLC is that it provides faster, more precise, and highly reliable results compared to some traditional methods. This speed and accuracy are crucial, especially when it comes to maintaining high quality in pharmaceuticals. Ultimately, UPLC plays an essential role in pharmaceutical quality control, helping ensure that products are safe, effective, and consistent. So, in the context of this presentation, you can see how UPLC is a key tool in modern pharmaceutical analysis, meeting regulatory demands while delivering on performance. For further support, please feel free to contact us. We're here to help you with any questions or additional information you might need. And with that, I want to sincerely thank you for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Remember, we're always available to assist you as you apply these insights. Thank you once again, and have a great day.